Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody back to In the Interim. Today I have a really cool topic. I was really excited about this. And the topic is a new kind of trial design. And we'll get into what's new about it, but the old version of powering the trial at 90%, running it and finding out was the trial successful or not, doesn't work here. So we'll introduce that in a minute, but first I want to introduce my guests. So two guests from Berry Consultants today. Liz Lorenzi, Dr. Liz Lorenzi, a senior statistical scientist at Berry Consultants, and Dr. Amy Crawford, a statistical scientist at Berry Consultants. Welcome both.
Thanks for having us.
So let's start with introductions. I was told that we started one of these podcasts and I didn't do a very good job of introducing our guests. So let's do a little bit of introduction. Liz, tell us, how long have you been at Berry Consultants?
I've been at Berry, I think 6 years actually. It started on July 1st, so I just hit my 6-year anniversary. Before then, I was at Duke doing my PhD in statistics. And since being at Berry, I've been really enjoying participating in lots of different clinical trial designs, all of which are adaptive, innovative, exciting. I have been a part of a lot of design teams for platform trials, so I have found a particular interest in working on the design of platform trials, which I think we'll be discussing an example of one of those today.
Yeah, very nice, very nice. And so, Liz, do you consider yourself a Bayesian?
Oh, I do think I'd consider myself a Bayesian. Okay. Yeah,
Okay.
I think I have to say that going to Duke for my PhD, but but I do think of myself as a Bayesian. Mm-hmm.
Yeah. And you'll get a flavor of that, I think, today as well. And Amy, tell us a little bit about yourself.
Sure. So I've been with Berry for just over 5 years. I started almost exactly a year after Liz. I've also been working on some platform trial designs, but I've also been working in stroke quite a bit. So, which I think sorry, Scott, teasing the topic. Going to talk a little bit about a stroke trial today. So it's a particular clinical interest that I've had since starting at Berry. Before I was at Berry, I was doing my PhD at Iowa State University, working on something very different. Not as Bayesian of a group there, but I think I also consider myself a Bayesian.
Yeah, and we'll see some of that flavor today as well. Okay, so the topic for today is part of the STEP platform. And a shameless plug for everybody to go back and listen to the podcast with Eva Mistry and Jordan Elm. We go through the big picture of the STEP platform. It's a fabulous effort, and it's a really nice potential of what platforms can be. And I think the NIH is actually a place that platform trials can be incredibly powerful. So let me not introduce this. And Liz, do you want to tell us just overall the STEP platform, the goal of the STEP platform?
Sure. So STEP is a platform trial in acute stroke, and this is funded by the NIH and kind of organized by a big group of stroke researchers under the umbrella of StrokeNet. And the goal of this platform trial started as in one of the areas of answering the question of what population does EBT benefit? So about 10 years ago, they learned that this therapy called endovascular therapy, it's a clot clearing device, was very effective in a particular patient population. And since then, there's been numerous questions of whether that we can expand that patient population and try to learn who EBT can benefit. And so one of the major questions under the STEP platform is to learn which patient population can EBT be beneficial for. There are other questions that SUP answers, more on the medical therapeutic side. So thinking of different other treatments that are a little different in the question of maybe more traditionally trying to answer, is this therapy better than a control? But I think the one that we're going to focus on today is this question of EBT. And it's a very cool structure to have a platform trial where kind of have this master protocol guiding the enrollment of patients, guiding the inclusion, exclusion, guiding kind of organizing sites and the infrastructure of the trial, and adding a lot of efficiency so that we can answer this question.
So in the STEP platform, it's structured so an individual, a team can propose a question to the platform. The platform has this larger master protocol aspect, and questions get asked. And a huge goal of this, as Liz described, is finding out who does and doesn't benefit from endovascular therapy. And we're going to say EVT over and over again. So this is the clot clearing device. Within the structure of this, when this question comes into the platform, there's a design team. So the design team is part of the master protocol, and it involves scientists, stroke neurologists, statisticians. So the three of us are part of this design team. There are many others within this design team, several others at Berry Consultants, several at Kansas University Medical Center, several individuals from there, MUSC at Jordan Elm with that. And so this is the design of that part within the master protocol. So in the setting where all trials historically were run, and they were really optimized. If you're running a trial and you want to show endovascular therapy is beneficial, historically they ran trials in those patients that were really, from their perspective, most likely to benefit from endovascular therapy. Relative to the control here is just background medical management without the device. And so they ran trials in that population that they thought were optimal. They powered it for that population and demonstrated endovascular therapy was good. And that's within a certain population. And it was incredibly effective in that. So now I think what neurologists and the medical community are stuck with is, okay, you're within this optimized population. This therapy is tremendously beneficial in the neurological status of patients 90 days afterwards. They're incredibly beneficial. So let's figure out in those we don't know if it's beneficial, whether it's beneficial. So now the first question came in, one we're really going to focus on, is we'll focus on the first question, which large vessel occlusions were generally thought that EVT was beneficial, but how about smaller size strokes? So can you set up a little bit of the patient population? And I know we're all statisticians here, Amy, but a little bit of the patient population that this question now is, does endovascular therapy work?
Sure, yeah. So like you were saying, Scott, there's this set of patients where we know the answer. And I like to think about it as you can sort of draw a box around that set of patients, right? You present with a stroke at the hospital, and based on how you present, the doctors figure out whether you should get EVT or should not. And that indication is relatively narrow at the moment. And so what we're looking at here is for smaller vessel occlusions. I think we're thinking about what we call medium vessel occlusions. What about patients that are just outside the box? What if you present to the hospital and you have a baseline characteristic of your stroke that doesn't quite fall inside the box, the medium vessel occlusion, you fall just outside the box, and the question is, should you get EVT or not? And that's kind of the question we're trying to answer. So these patients, where we're trying to answer this question for, they have a little bit sorry, their strokes are a little bit different than those that are part of the indication. And I'm going to fumble the clinical piece here.
And that's okay. Well, you get a lot of leeway in the clinical piece. Yep.
So there are a couple baseline characteristics that we look at for strokes when we're trying to answer this question. One of them is time last known well. So patients who present earlier after they were last known well with their stroke, we think that maybe they benefit more from EVT in this population than patients who present later. And so the question is, what should we do for patients that present later? And there's another baseline indicator called the NIH Stroke Scale. And so on this NIH Stroke Scale, you present with a stroke and you get a score. So certain patients with certain scores are indicated for EVT in this population with medium vessel occlusions, and certain patients aren't. So the question that we're trying to answer for these patients is, what kinds of baseline scores on this scale should we give the therapy to?
Okay, so let's set the problem up here. Is we're now going into medium vessel occlusions. We're enrolling people. I know papers have come out that have changed this, and so we're updating the design. But let's go back to the first version of the design for simplicity, where it was medium vessel occlusions, and the thought was that it's the relative efficacy of EVT compared to medical management is very likely to depend on the clinical symptoms, which is the NIH Stroke Scale. So this is a stroke scale from 1 to 30, say. I think it actually goes a little bit higher than that, but those are very rare. Was it 28 maybe? That this scale. So this is their range of symptoms, and really the size. This is a proxy for a medium vessel occlusion stroke. The clinical symptoms are maybe telling you how much brain there is to save through endovascular therapy. Now, all of these patients are within 24 hours last known well. So the statistics problem here, the trial design is, we want to find out who in this range is a function of their stroke scale from 1 to 28 benefits from EVT or not. Now, what would be wrong, Liz, with us running a traditional trial and saying, let's enroll everybody in there and do a test whether EVT is beneficial in that population?
Yeah, so, we believe we know part of this population, from these previous trials benefit. If we just say, let's expand, run a giant trial, randomize 1-to-1, and at the end test, we would likely get a null result, because likely there is heterogeneous treatment effects within this population where some are going to benefit, some will likely not benefit, and medical management might be better. And so what we really want to do is try to learn, based on patient characteristics, which patients are benefiting from EVT. So we think about this NIHSS as one of the covariates we could use to try to stratify the patient population. We might want to learn within each bin of NIHSS what the treatment effect of EVT might look like. But the question isn't necessarily what is the treatment effect in the bin, but it's really, is EVT better than medical management? And it's more of a generic question of, which of these bins should benefit, which of these bins may not benefit. So it's really kind of learning where that change might occur in the patient population.
So you said the word change, which is going to be a key word. Maybe not jump to change quite yet within this. So if we enroll this entire population and we run a test and find no result, and we might say, in this population, there's no benefit, where there may be people in that population that benefit a great deal. So the mathematical problem here is we have a scale from 1 to 28. And we're trying to find out for those patients who on that scale benefits and who doesn't. What somebody could do to attack this problem, if we're stuck in the old way of running a clinical trial where we run a trial and we test a hypothesis, and it's only upon rejecting the null that we say, oh, EVT is beneficial, which were done in the first ones. We might go at this and say, let's just take the big ones. And this was largely when people wanted to originally show it was beneficial, they did that. They took the big strokes. And so let's take everybody that's 20 above and test to see if EVT is better in that scenario. And then if that's successful, maybe then we go down to 15. You can imagine how slow science would move if we're restricted to run these yes-no questions in the trial. So we're going to do something different in this trial. And we are going to enroll everybody in this patient population. And we're going to try to estimate who's better and where does EVT work or not. Now, we're going to use the scientific information that on the bottom part of the scale, that medical management is likely to be better. On the top part of the scale, EVT is likely to be better. And somewhere there's a change point here. So tell us about doing this with a change point model in trying to estimate that.
The idea here is that we want to figure out where to draw the line, who should get EVT, who should not. And so the idea is if we enroll everybody across this scale where we don't know the answer, we can let the data inform where the line should be drawn. And we do this in a structured way because, as Scott said, we know that on the higher end of the scale, somewhere out there, EVT is beneficial. And the question is, how far down can we drag that line down the scale to where EVT is still beneficial for patients that lie above the boundary? And we let the data tell us what that looks like. And the way that we do this, like Scott said, is with a change point model. And what the change point model does is it looks for that single point of where we draw the line, where we draw the boundary to expand the indication of EVT along this baseline covariate scale. And the data inform that. And so the really beautiful thing about a change point model is it allows us to make these informed decisions in a structured way. So we don't have to test, you could think, maybe if you're coming at this for the first time and you want to say, oh, does it work in bin, 28? Does it work in bin 27? Does it work in bin 26? You can think about, well, let's test it in bin 28. Let's test it in bin 27. Let's test it in bin 26. It's going to be another extremely inefficient way of doing this, right? And so using all of the data and the neighboring bins inform each other, all of that data to figure out where the single point the line should be drawn is just a really nice framework for how we answer the question.
So within the model, if we learn and we get a lot of patients near 15, 16, and EVT is doing better, it tells us the change point is to the left of that. And 20 is also better. 25 is also better. So in some way, we're trying to figure out this one place where it shifts from medical management to EVT in the setting. Now, we use a Bayesian model for that. Not surprisingly, I'm talking to two Bayesians here. They wouldn't let me get away with a frequentist model here. So we're using a Bayesian model in this setting. Now, Liz, it didn't really work when we had a single change point. And, we thought of that break point of where it goes. Why didn't a single change point work?
Yeah, the single change point assumes that to the right, there will be a benefit of EVT, and to the left, there will be a benefit of medical management. And what we were learning was that it might be a little bit more nuanced than that, where there might be a subset of patients where there's essentially no difference of the two therapies. It's essentially like a coin flip. And so by changing it from one change point to two, we're allowing ourselves to kind of find this period or this area of somewhat equality or a similar effectiveness. And then we're allowed to say to the right of that, that's where the EVT would be. So to the right of the right change point, that's where EVT is better. between the right and the left change point, they're equal. And then to the left of the left change point, we think medical management is better. So it allows us a little bit more flexibility to add that kind of piece where we don't actually think there's a big difference between the two.
So we were simulating and we discovered a bit of the issue with one. And in part, it was the old bugaboo for us, the null hypothesis. So if you simulated a scenario where it's flat across the entire stroke scale, the model doesn't really work well. And you can imagine it's almost non-identifiable that the change point could end up any one of these places. And maybe it ends up right in the middle of that. And it somewhat is misinformed by saying it has to be positive on the left and negative on the right. I think I said that backwards, but that it's beneficial on one side or the other when there is this range. So when we simulated that, we found it really didn't work very well. So we created two change points, Amy.
Yes.
Yeah.
Yes. Yep. So we created two change points. And what happens then in the null scenario that you were just describing, Scott, is it allows those two change points to scoot away from each other if that's what the data are saying should be the right answer, and allows for that region of equivalence or equality that Liz was mentioning that we would estimate between the two change points to cover the entire scale of that covariate that we're modeling over. And so it allows us to handle this null scenario much more intuitively than if the model were to have to just try to figure out where a single change point would need to be and the data look the same across the whole thing.
And then we really care about the place where EVT is better. If they're the same, you wouldn't do this invasive device. So it's somewhat of a statistical and clinical better fitting of the data, and it functions much better. Another value of clinical trial simulation to carry out a range of scenarios, even if we don't think it's very likely, and we've created a better model in that. Okay, tell me a little bit about the code. I think Amy has carried the brunt of the code building here. Now, can we just create our code to run this, to run simulations? That didn't really work.
Yeah, that didn't really work. So when we write code in R or one of these other languages, what we do when we run Bayesian models is we tend to use some built-in packages. Statisticians listening have probably heard of Stan or JAGS. And these built-in packages don't necessarily facilitate learning the type of model that we're describing here. And so we've had to create custom code. We wrote our own sampler, our own code that fits this model to run custom for this trial.
So, we wrote that in a lower you wrote it. You wrote it in a lower level language. You wrote it in C to then be called for simulations and within that circumstance. And it's a Bayesian model using Markov chain model where the unknown is the location of the change points within that. Beautiful, beautiful code. And I'm ignoring a good bit of work to get to the right place and all of that, which was fun in that. So now we're running the trial design in this circumstance is where we may, as the data start to come in, we don't want to enroll 2,000 patients and then analyze this. So that's the model that will be fit, but what do we do with the design? So what's the design that goes with that model, Liz?
Yeah, so this is an adaptive trial framework. So we, through the STEP platform, will have interim analyses that occur at that time. We would gather the current data, run the analysis model, and then evaluate against some pre-specified decision rules to just see if there's any conclusions that could be made. And what's nice about this is, we have a lot of information on the edges, like we've talked about. So we may not need that many patients to inform those bins that are, right outside that box that Amy described. And so by doing these adaptations, we may be able to get some results and conclusions out more frequently, change practice, facilitate, better learning of this question through these adaptations. So I think we have them set to be done quarterly. So as we're enrolling the trial every quarter, we would gather the data, run the model, and that would be done by a blinded team, or an unblinded team. So we will have a separate team from the three of us, or the Baker Berry design team, that will be unblinded to the data and firewalled from us on the design part. And they would be the ones that get the data, run the model. And if there are decisions, they could then make a public announcement through the DSMB, et cetera. So I think that's currently the way that we have this set up.
So this is really cool in the way of we've got this model, and as patients come in, it's a wider frame of we don't know who EVT benefits and who doesn't. But as soon as we learn that, we stop enrolling that group, and we announce that for patients medium vessel occlusion with NIH Stroke Scale above 18, EVT is beneficial. We publish on that. And meanwhile, those patients aren't enrolled. And we're narrowing in across the scale on who should get each particular treatment. And one of the things that was wonderful when we were doing simulations of this and we're characterizing how well this worked is it's not just about power, but what fraction of the patients when the trial's over are we going to treat well? And how are we going to treat patients when this trial's over? Across the scale, now we might give them medical management, we might give them endovascular therapy, we might give them the wrong therapy that they would have benefited or they're harmed by it. And we can create scenarios of truth, run this trial, and then evaluate how are we caring for patients when the trial's over, which is such a cool operating characteristics and so targeted to the trial. So that was, there were many of these, but that was one of the cool things we evaluated in the simulations. So science pulled a bit of a switcheroo on us, and this is a very active area of research, not surprisingly. And so new trials have come out where medium vessel occlusion trials have come out really questioning whether EVT is beneficial. But it has given us new information in this population that a key parameter is very likely time last known well. How acute is the injury is probably related to endovascular. So now this has become not one dimension of NIH Stroke Scale, but two dimensions in it. And we have a new submission to STEP, and I don't know how public that is, so I won't describe the clinical context, but that one is also endovascular therapy versus medical management, and it's within the realm of the patient population we don't know, which is also two-dimensional. And it's the same structure that on one side of the scale, we know EVT works. And in the other side, we know at the very extremes that medical management is better off of the patients we're enrolling. So we know that direction in two directions. So this is, we know up and to the right is one treatment and down and to the left is the other one. We just have no idea where the break is in this. So now the model's gotten a little bit more complex, Amy.
It has, yeah. So our model now actually has 4 change points. Like you kind of described, Scott, we're working on a grid. And so across NIH Stroke Scale, we're trying to figure out within patients who present early, it's less time, last known well, maybe they should be treated differently than patients that present later. It's been a longer duration since their time last known well, since they've had their stroke. And so what we're doing now is we're trying to draw lines on a two-dimensional grid instead of a one-dimensional baseline covariate. And the way we do that is with 4 change points, and they're restricted and they move around with each other. But I think the big kicker with this more complex framework is that it follows clinical belief and knowledge, and we've been able to adapt and update what we're doing based on what is being presented and changing practice. And so now we're shifting our approach, and it still is going to follow this structured, data-driven decision-making framework, and the model matches the decision framework that the clinicians and the medical world, would expect to see based on these recent findings.
Yeah, that's so cool. And now, Liz, you are going to jump into one where it's not necessarily 2 change points, but it's a change curve, perhaps in 2 dimensions. And so this idea was presented just this week, that we have a 2-dimensional space. Think of quantitative variable on the x-axis, quantitative variable on the y-axis, and we know what's in the upper left and we know what's in the bottom right, and there's a curve that we now have to fit. And there's monotonicity across this. So you're jumping into a pretty cool problem with a bit of uncertainty how this is all going to behave.
Yeah, and I think what we learned from Amy's, development of the original change point model is that this really needs to be simulated, and it needs to be discussed and, constantly put in front of the clinicians to understand what they're thinking and what they're expecting and how we can make sure the model aligns with that question. I'm not sure if I know exactly what that model is going to look like, but I think, as we continue discussions, we'll have a clearer picture, and simulation will be our friend in these conversations to make sure that we get everything right.
Yeah, we would be incredibly fearful if we just kind of put a model together and the first time we use that is in the actual trial. So we simulate millions of data sets. We see what it looks like under a range of scenarios and make sure it functions well, because we don't want the first time we use it to be the real trial. We want that to be the 10 millionth and third time that it's used. It's just been used on the simulated data set. So we will drive you crazy, Liz, by test driving this over and over again. But it's sort of the fun of science here. All right. So while they are enrolling, actually, I believe there's a bit of a pause in enrollment in this setting. So we are actively working on this. The STEP platform is a very cool platform. By the way, if that kind of modeling interests people, there's another NIH trial, IceCap, that just stopped enrolling that has analogous models. It's a different model, but has an analogous model, and that will be reading out sometime in the next few months if that's kind of interesting. And then hopefully we'll get STEP results out. So Liz and Amy, thank you for joining us in this weird place in the interim and sharing this really cool modeling.
Yeah, thank you so much for having us.
Yeah, thanks, Scott.
Appreciate it. So everybody out there, thanks for joining us here in the interim. Until next time.